Okay, continuing with this. Amide is also related to this. You can see this RCO. Remove that OH, get this NH2 kind of a thing. So you can think of amide being a combination of an amine and a carboxylic acid. Right? What is a nitrile? It will become important in origin of life things. So there are important implications of nitriles. Okay, what will be isonitrile? So whenever you have an iso prefix, so iso generally means what? Same. In here, that doesn't hold. I don't know why they use this, I should look into it, but this becomes RNC. Okay. You guys have to tell me, how is this possible? How many bonds can nitrogen make? 3. How many bonds is it making here? Think about it. I get this. Carbon is making 4, nitrogen is making 3. How is this happening? In here, carbon is making 3 and nitrogen is making 4. How is this possible? Think about it, okay? The isonitrile thing. Okay, with this, let's focus our attention on some of the molecules that we see in biology. Okay, so in what we are trying to study are biomolecules. So those kinds of molecules that are essential for life. Okay, what are the building blocks of life? So typically you will look into these things: lipids, carbohydrates, proteins, and nucleic acid. All of these are polymers. What does poly mean? Many. Right? So they are formed of many monomers. So when you join monomers, you get polymers. Okay? So if you imagine, like if you have a compound like this, imagine this having 2 hooks of some kind. Okay? One is this upward and one is this downward. Not necessarily, I mean, this is a representation, okay? So this, they don't really have a hook. Okay? So this will be a monomer. If this is a monomer, my polymer will look something like this. So you can have one of these. There will be the next one will be on, maybe I'll just make it a little. So the next one is going to come in here and clip it on this, right? And the same thing will continue. Okay. And you can just go on and on with this. Okay? So monomers are, when they come in together, they form polymers, and you can think of these things as the linkages. And we'll see what are those chemical linkages with which you can actually make the polymers. Okay? Now, let's start with proteins. So, amino acid and proteins. Amino acid is the monomer, and when they combine, they get the protein. So this is the polymer. Now the basic structure of an amino acid is something like this. You have an alpha carbon, as we call it. So an alpha carbon is just an sp3 hybridized carbon. That's what an alpha carbon is. So think of it as that one. And there could be many sp3 hybridized carbon, but the first one is typically called the alpha. The second one will be called beta, and so on. So the first one, which is the most important one, but it has to be a sp3 hybridized. So how many total bonds? There will be 4. One of them is always hydrogen. It's very convenient. One of the linkages is always hydrogen. The other one is always an NH2 group. The third one is a what is this? The COOH? The carboxylic acid group, right? And this one is an R. So the COOH, H and NH2 are always fixed. But this can vary. So this in the amino acid is the main thing. Rest everything is just, this can vary. Now, when this R becomes hydrogen, what will be the formula for this thing? What will be that compound? How will you represent that compound? NH2. So this and this both this and this is hydrogen, NH2 and COOH. This is the do you think is the simplest amino acid that could be? Because what else? You cannot go more simpler than this, right? You have to at least have a hydrogen. So this is the simplest amino acid. This is called glycine. Now try to remember chirality. And tell me whether this compound is chiral or not. What was the definition of chirality? Or how would you find the chiral carbon? What was the definition of a chiral carbon? Look into your definitions. What was the definition of a chiral carbon?
All the different species will be attached to it.
To what?
The carbon.
What carbon? Chiral carbon is first of all an sp3 hybridized carbon. It has to start there. You have to start looking for an sp3 hybridized carbon with 4 different groups. So there is only one sp3 hybridized carbon here, the central one. Do you think this carbon is chiral or non-chiral? This one. So there is only one carbon that is sp3. This is sp2, right? So this is hydrogen, hydrogen, NH2 and CO. So this is only one carbon, this alpha carbon that you have to see. Is this chiral or not? It's not, because it has 2 same groups, because this could be the plane of symmetry for this molecule. So this is symmetric, so this is a non-chiral carbon. Interestingly, this is the only non-chiral amino acid that is there. Rest all amino acids that you will see are all chiral. This is the only one. So every time you see an amino acid, you will see that people are saying it is either a left-handed or a right-handed. It is a D or an L amino acid. It's always we say it's either a D or an L. Okay. Now there are about 500 amino acids known. So you can see, so when the R becomes a CH3, what will you get? NH2, CH, CH3, COOH. This thing is alanine. You just change that R to a CH3. Okay. Now let's change it to CH2OH. So this is serine. If you just take this out and remove one hydrogen and attach a benzene ring to it, this becomes phenylalanine. It's one word, phenylalanine. And so on and so forth. What you can see is this R has the capability of controlling a lot of the chemistry, or at least some of the physical properties. The main chemistry will be happening in this ring, right? In here, the main chemistry is going to happen here. This is the side chain. This is the side chain. The main seat of chemistry is this. This is the side chain. But the side chain can impart a lot of the properties to the amino acid. Now, for example, I can have an amino acid with 7 different carbons in here. It can be really complicated, big. And what do you think the solubility of alkane is in water? Do you think alkane? So if you take the hydrocarbon that you fill in your car, if you take a little bit of that, it's a liquid, right? Although we call it gas because it's a misnomer. It's a gasoline is the short for it. It's not actually gas phase. It's a liquid thing. If you take the petrol that you are putting it in your car and put it in water, will it be soluble or not soluble? What's your intuition? What have you thought? What have you heard? It's not soluble. You can try. It's very similar to an oil. So oil and water separate, right? So those big carbon groups impart a hydrophobic character to the molecule. So if you have long carbon chains, these are all giving it a hydrophobic character. So these won't dissolve in water. So if you have this big group attached in here, what do you think that amino acid solubility will be in water? It will not be soluble. Now, the bigger the group is, the more difficult it will be to dissolve. Now, do you think what happens to serine? Will this be water soluble or not? What does this look like, this group? What is the character of this group? It is looking like an alcohol because this is this OH group. And what is the solubility characteristics of alcohol in water? Are they soluble or not soluble? They are soluble in water. They are perfectly soluble in water. Why? Because of the OH group. Why? What does the OH group do? What is the impact of that OH group? Why is it making it soluble? It is because of the OH group. But what is the OH group doing? No dissociation. I am not asking the acidic behavior of acids, acidic behavior of alcohols. I am asking the solubility behavior of alcohols. What controls the solubility of alcohols? Why are alcohols so soluble in water? What is that? What is that thing that is controlling it? Hydrogen bonding. Why is water so remains as liquid? Hydrogen bonding. And this is the OH that will now pair up with the waters. That is why alcohols are more soluble in water. And so RCOOH, carboxylic acids, they will be soluble in water. So this, what do you think serine will do? Will this be soluble in water? Will this amino acid be soluble in water? Yes, this will be soluble in water. But if you have a really big group, right, like, so even simple groups, the alanine, so if you just have the CH3, so forget about this, if you just have the CH3 group in here, if you just have the CH3 group in here, the alanine is water insoluble because it's that CH3, it's hydrophobic, it's not allowing it to dissolve. But remove that H and add an OH, it becomes soluble. So this side chain controls the properties of the amino acids, right? So you can have different kinds. So there are about 500 known amino acids. Out of these 500, only 20 amino acids are known to be important for life. So for life, we only need 20 amino acids. We don't need 500 amino acids. So life is very selective in that way. Or the other way of looking at it is this, that there is no pressure on life to use all the 500 amino acids. Life doesn't need to use everything that is there. No, it does not. It will be selective. Maybe the selectivity is because of the solubility. Maybe it is because of availability. Maybe it is because of the need, the function of that particular amino acids, whatever. But you actually need only 20. I might be wrong, but I think 18 or 19 out of these 20 are all left-handed molecules. And I said, all these are handed, all these are chiral, except glycine. So I think 18 of them are left-handed, one is right-handed, and one is just non-chiral. So that is a particular signature of life. So if you have, say, R, I don't know which one you need in the R form, but you can have the right-handed form of that molecule, and life will reject it. It will not use it, or it cannot use it. We don't know the reason, or at least I don't know the reason why it is left-handed. Why do you need left-handed molecules for life? I don't know that. Okay. All right. Now, coming back to the basic structure. So you can have all these different things happening, right? But coming back to the basic structure again. At neutral pH or at normal pH of body, which is around this neutral, what happens is this COOH, so hopefully by now you know that this H is acidic H, right? Because this is attached to an O. So this H is an acidic H, and that's the seat of this acidity in this carboxylic acid. So this H is acidic. So what can happen is it can lose this hydrogen. So you can get this hydrogen, H plus, floating around. Who will take this? Who reacts with an acid? A base. So where will this go? To a base? Can you see a nearest base? So what is an acid? Proton donors, right? So that was the basic definition. So here is proton donor, right? There are also other definitions of acids. Anything that reacts with an OH minus is also acid. But you can also have Lewis acids. What's a Lewis acid? Electron acceptors are Lewis acids. So by that definition, what is a Lewis base? Electron pair donors can be termed as Lewis base. Can you see an electron donor in the molecule? In the same molecule that produced this acid, is there a seat in the molecule itself that can act as a base? This N, right? So a molecule, see all these amines, these are bases. And we'll use this more and more, multiple times in biochemistry. These bases. What is the seat of that basicity? These electrons. Now, some of them might be readily available, some of them might not be readily available. So the amount of, or the propensity of the basic character will change, but nevertheless they're all basic. So we'll see pyrene, pyrimidine, all these kinds of bases, and we'll see them in the DNA structure. So these play a very important role there. In here, this H plus is taken up by these NH2 because this is a base. What happens is this H plus comes here, and this becomes a plus. So this molecule is an acid and a base both by itself. So it has an acidic center and a basic center. And this can exist as a neutral molecule overall, but has a plus charged end and a minus charged end. This is an ion, but this is neither a cation nor an anion. This is a Zwitter ion, in which both of the plus and the minus are on the same molecule. Now, a very important thing is going to happen here. So till now we were looking into the monomers. Let's form the polymers. So, for example, we can have this NH3 plus, say you have this H, this R, this COO minus group. You can have another amino acid, another monomer come in here, and this will be N, C, H, maybe this is R dash, maybe this is R, doesn't matter. What will happen is there will be a bond formation here. With this O minus and this H2 or H3 plus in here, what will happen? There will be a removal of a water molecule. And you will form a carbon and a nitrogen bond. It's not that long, but this is the linkage. This is the linkage that we were talking about between two monomers. So now you have started to make a polymer.
There will be another H in nitrogen.
Yeah, H. This is called a peptide bond. The name of this bond is called a peptide bond. And we can call this the polymer made from the amino acid with the peptide bond. We can call it a polypeptide. Also a protein. Now this is a very simple arrangement, which is, we call it a primary arrangement, okay, or a primary sequence in which it goes, you're just forming these polypeptide bonds in a chain. So you can have hundreds of these things, one after the other, and it will still be a primary sequence because you're just adding one thing to the other. When things get convoluted, say all these chains are coming in and interacting on all that, then you start forming secondary. Even that can twist and turn, that can become tertiary, and so on and so forth. I don't know if you guys ever saw like base phones. Like, did you ever see the phones which used to ring with the wire and the receiver and all that? That used to have a coiled wire. So imagine that you are making a wire, which is the primary sequence. That wire is getting coiled or interacting. That becomes a secondary. And even that can get twinkled sometimes, right? That becomes a tertiary, and so on and so forth. So proteins are these polypeptide bonds, but different layers of complexity. But this is where it starts, okay? Very simply, I'm not getting into all that, but understand that this is what amino acid is. This is how you form polypeptide proteins and stuff like that. Proteins are very important, right? So they can act as catalysts in biological reactions. We call those amino acids that act as catalysts as what? What do you call those proteins that act as catalysts in biology? Enzymes. So anything that has an enzyme, it's a protein of some sort. Anything. And you know that it will have all these